I can't find the button one, so I have to first share my screen. Right, yes, so thank you so much for inviting me to come and give the seminar. It's so nice to give a seminar in person for a change, and also to come to France. This is my first time in France, so really loving it. Thanks so much. So like Simon was saying, I'm a large mammal ecologist from South Africa originally, and I've only recently started in Denmark at University of Aarhus. And generally I work on aspects like trophic impact and trophic interactions and how animals are impacting ecosystems through disturbance and through feeding and so on. But one of the things that I've been quite interested in lately in the last couple of years now actually, but is consumer-driven nutrient impact in savannas, and particularly how the diversity of species could modulate these kinds of impacts and modify them. And I don't consider myself a zoogeochemist. I'm leaning considerably more to the zoo side, not the chemistry side. But these are definitely topics that I see myself working in the future and developing it. And I'm definitely not the only one that's interested in this. Because over the last couple of decades, you've seen so many studies that's come out in zoogeochemistry, and particularly in terrestrial zoogeochemistry, which is relatively new compared to the lake systems, for example, where people are going out and trying to see how in their system, animals are acting as these sources and sinks and vectors of nutrients, and thereby kind of shaping nutrient landscapes. And in the beginning, I think especially the starting studies, the illustrative studies of terrestrial zoogeochemistry was very case study specific. So people were taking these particularly idiosyncratic species and looking at how they were influencing nutrients. For example, like if animals are moving nutrients against an abiotic gradient, like anadromous fish, like salmon, moving it upstream, or where animals are able to take nutrients from one ecosystem and transport it into a different ecosystem, and kind of crossing these boundaries that would otherwise be really or less permeable, I suppose. Like, for example, if bears then catch those salmon and distribute to a terrestrial system, or hippos in Africa that's feeding terrestrially and then defecating in the water and supplying these nutrients to fish populations and diatom populations and so on. And these kind of idiosyncratic studies where the animals have had this very distinctive impact on nutrients have, of course, been very informative, but I think one of the greatest values has been that they have this immense fascination value, and it's really spurred people on to go back to their own systems and think about how their own organisms and their own ecosystems are impacting nutrient landscapes and shaping nutrient landscapes. But of course, if you have a bit more of a systems perspective, then perhaps you'd be interested more in the net effect of communities as a whole. And how do all the different species that make up this community, how do they interact to create nutrient landscapes? And that's where our interest lies. So my co-authors and I, we work on aspects like trophic ecology and species-specific traits like body size or body stoichiometry, how that influences this consumer-driven nutrient impact. And from the pictures that I've used, you probably have deduced that we're focused on large mammal communities, and particularly in African savannas. But African savannas, when you're working with large mammal communities, African savannas are very nice platforms to work in, because they've got all of this diversity in herbivores and in carnivores, and they've got herbivores that range in body size across multiple orders of magnitude. So it's quite a convenient system. So what my co-authors and I have been asking ourselves is that if you work in these diverse communities, if we're talking about diverse communities, how does the herbivore community shape savanna nutrient landscapes when you've got all of these different species potentially all doing their own thing and perhaps having contrasting effects on nutrient landscapes? So we've been working in this savanna reserve in the east of South Africa called and Pelosi Park. It's a very interesting system to work in because it's one of the oldest systems in protected wildlife reserves in Africa. It's experienced some form of formal protection now for 120 years. The other thing that this park is known for is that they were key in saving the white rhino from extinction in the beginning of the century. And the reason why that's relevant for this work in particular is because you've had this very, important keystone species where the ecological impact from the species has gone on relatively uninterrupted for the last 120 years in Fifuet. The other really nice thing is that despite that it's a relatively small system, Africa-wise, it's only 900 square kilometers in size, but it has a huge variety of herbivores and carnivores, and it contains functional densities of mega herbivores. And with mega herbivores, I mean like the functional category of mega herbivores. So the generally things larger than 1,000 kilograms. So they're functionally distinct because they are their populations cannot be controlled by predation. And then the last thing that I would want to point out that's particular about HIP is that it's got this very stark rainfall gradient with about 550 millimeters per annum in the south and almost 1,000 millimeters per annum in the north, even though this distance between is only about 50 kilometers. So that means that there's this huge vegetation type turnover, and there's a lot of vegetation heterogeneity in terms of habitat type, but also in terms of biomass buildup. And I think what's also characteristic often of these mesic savannas is that you have a lot of even the fine scale vegetation heterogeneity and vegetation structure. So you can find quite dense vegetation areas that then open up into these gaps that quite abruptly. And when you find these gaps, they often look like this, where you've got this immense amount of fecal nutrient accumulation. And not all of them have a watering hole, but a lot of the gaps will have this massive amount of fecal accumulation. And of course, we're considering that these animals are congregating in these gaps as an antipredatory response. So the gaps are, because they're open, they're more visible, and the animals get to see further, and they get to, see a predator coming and respond in time. But this addition of urine and feces, of course, is a huge nutrient input, and this could lead to local soil enrichment. But also in these systems, the susceptibility to predation is a function of body size. So you have these mega herbivores that we would expect would be too large, too difficult, too dangerous to hunt regularly. So they should not be responding to the risk of predation as much as the smaller species are responding. So we wouldn't expect these large animals to be congregating in these gaps. So what my co-authors and I have been asking is, to what extent were these mega herbivores indifferent to predation? And how, if so, what did this indifference mean for the redistribution of nutrients across the country? All right, so to get at this question, we set up an experiment in Fifuet and Pelosi, and we tried to create artificially these gaps in which animals can congregate. But we created them in such a way so that they would differ in the surrounding visibility, so that they would also represent this gradient in fear, or perhaps rather a gradient in the perception of how animals perceive risk in the environment. And we did this by clearing plots within, are you seeing that? No. Oh, no. Okay. So we cleared these plots in the like a more dense savanna matrix. And then we either left the surrounding woody vegetation intact to provide a lot of ambush opportunity, or we also then cleared some plots of woody vegetation in the surround to take away that ambush opportunity and presumably make it quite a bit safer. I'm so glad you have a stick and not that little red pointy things.
Yeah, though there are more people online, so
right.
They might want to also see.
Do you have a red pointy thing?
Oh, you can use it.
Oh, of course, yes.
Can use both.
I can do that. So no, I just wanted to point out that you have, so you can see the difference that the contrast that we created here. Of course, the top one is the uncleared plots, and the bottom one is the cleared plots. And we ended up with 12 plots in total, so 6 cleared and 6 uncleared. And we then put up a camera trap and we monitored species vegetation and fecal accumulation over a period of 2 years. All right, so this is what we found. So just to place you in these graphs, so these the first column is for risky plots, the uncleared plots. And then the next column is the safe cleared situation. And the top row is for nutrient accumulation through feces, and the bottom row is a measure of the time spent on the plot, which we used as a measure of herbivore impact. And the picture of the rhino represents the mega herbivore impact. The zebra represents the meso grazer impact, so grazers less than 1,000 kilograms. And the kudu is the meso browser impact. All right, so as we expected, the meso grazers ended up spending a lot more time in the cleared plots, in the safe area. And they really focused their foraging effort in these safe areas. And importantly, they deposited a lot more fecal nutrients within the safe areas compared to the risky uncleared plots. And it might be a little bit difficult to see those numbers, so I'll just point out that in the cleared plots over the period of 2 years, cumulatively speaking, over all 6 uncleared plots, the risky side, we recorded 8.9 kilograms of fecal material accumulation. And this compared to 24.5 kilograms of fecal accumulation in the cleared safe environment. In contrast, the mega herbivores, they ended up using the landscape a lot more evenly, and they also defecated a lot more evenly. So they, in fact, tended to focus their foraging effort more on the risky, uncleared plots, but this was not a statistically significant difference. They would rather just ignored this differences in visibility and presumably in risk that we created, and they just used everything relatively equally. But as you can see, their fecal nutrient return was really, really low to both the control and the treatment plots, because where the other species, the smaller species, were really congregating in the open areas and spending a lot of time there. They were spending nights there and resting and ruminating and adding more and more nutrients. The mega herbivores, because they were just using this without regard of the visibility differences that we created, there was no accumulation effect, so no congregation, no accumulation. So we realized that we need to stand back a little bit and broaden our perspective to more than just these relatively small manipulated plots. So what we did is we quantified fecal nutrient input for all the herbivore species, the large mammal herbivore species across the entire park. So we mapped the dung piles walking these transects that you can see here, and the transects covered practically the whole park. That southern area that you see uncovered is it's a wilderness area, so it's not so accessible. But in total, we walked about 190 kilometers of transect, recording where we found dung piles, who was responsible for the defecation, and in which habitat type it accumulated. And what this data shows is that when you compare habitat types that are very different in visibility, so for example here the open woodland, that is a very visible habitat type compared to a thicket that is very dense vegetation, low visibility habitat, then you can see that the meso herbivores, overwhelmingly their fecal material were found more often in the open visible habitat than in the thicket less visible habitat. Whereas the mega herbivores showed the opposite pattern. So their fecal material accumulated much more in this thick habitat, dense habitat compared to the open woodlands. So what this shows is that there seems to be this countercurrent of nutrient movement that is indirectly powered by this differential fear of predation, where you have the smaller animals congregating in these areas of safety, in these gaps, and presumably adding nutrients to these areas, importing nutrients, maybe accelerating cycling rates. But the mega herbivores use the landscape a lot more evenly, and they distribute their nutrients a lot more evenly. So the fact that they're using both the open areas and the closed areas, the risky areas, means that they're providing this kind of avenue along which nutrients can move back up this gradient that was created by the fear of predation. The other thing that we noticed when we got to this point in the study, looking at this little schematic summary, is that the fear of predation to some extent is influencing the identity of the defecator. So if you're a plant that happens to be growing in this risky side of the landscape, then maybe you'd be quite dependent on these big things like rhinos and elephants and so on to provide any additional fecal fertilization. So we were asking ourselves, does that matter? Does it matter whether the animal doing the pooping is an impala or a rhino? Does the identity of the defecator, does it have any nutritional, ecological, nutritional, ecosystem nutritional consequences? And the reason why you might suspect that perhaps it could is because animals should different sized animals should differ in their nutrient requirements. So, and if they're eating the same diet and they're taking different ratios of nutrients out of the food because they require them in different ratios, then what remains in the fecal material will differ along the body size axis. For example, so nitrogen is an important component of protein. And so maybe you would expect that the requirement for nitrogen would change with body size in the same way as metabolic rate would change, which is with an exponent of 0.75. Phosphorus, on the other hand, is a really important component of bone. So, and it's been suggested before that the allometric relationship between phosphorus or between skeleton size and body size is with an exponent of more than 1. So basically, as the animal becomes bigger, the skeleton becomes disproportionately bigger. And if this is the case, then it means that a larger animal should require less nitrogen and more phosphorus compared to per unit body mass compared to a smaller animal. And if they are eating the same food, which we'll get back to in a minute, if they are eating the same food, then it means that the fecal material of larger animals should have higher nitrogen to phosphorus ratios than the fecal material of smaller animals, because that's then what remains in the fecal material after they removed whatever they were able to remove. So we wanted to explore this question a little bit more. And again, in Terrestrial Pseudochemistry, and again, we used a particular characteristic of Terrestrial Pseudochemistry that makes this park such a nice system to do savanna ecology research in. And that's the fact that it contains a huge, like a large density of grazing lawns. And these grazing lawns are these very noticeable short grass patches in the savanna, and then they're grazer maintained, but they're not short because they were just eaten. It's an entirely different community of grass, solaniferously growing or rhizomatously growing grass species. And it's a very convenient system to look at the trophic impact and the control that herbivores would have on plant resources, because it represents this really tight link between grazers and grass and nutrients, with a kind of a circular dependency, because the stoloniferous grass are really dependent on the grazing pressure. They really require grazers to come back and keep eating them back down, because what happens is without the grazing pressure, you've got all these tuft grasses moving into the lawn and taking over and then out-competing and out-shading the stoloniferously, the short-statured grass. So they really require this heavy grazing pressure, and they can tolerate a lot of grazing pressure, partly because they are so short-statured, so they're quite protected from bites and the loss of like meristematic tissue, for example. But they attract a lot of grazing because they have this very strong compensatory growth response. So as the animals are eating them, they're just growing and growing and growing and providing all this extra fresh growth that attracts the animals back. So in summer, when these lawns are actively growing, you see all kinds of sizes feeding on these lawns. You've got from impala to rhino all congregating on these lawns. And then, of course, because they're congregating there, they have this huge fecal nutrient input, which is what is believed to sustain the compensatory growth. So there's this very tight link between, grass depending on the herbivore grazing and herbivore grazing, obviously depending on the grass and supporting the grass growth. All right, so we selected 15 of these grazing lawns that occurred all over the park. All right, it occurred all over the park, and we selected them in such a way that they differed in the surrounding visibility, so that we would again have this gradient in risk and a gradient in fear, gradient in the perception of risk, or what should I say? And as with the experiment that I was talking about before, we set up a camera trap and we monitored herbivore use and dung accumulation, this time for a period of just over a year. And we also went and got soil samples and grass samples, and we measured, analyzed them for nitrogen and phosphorus. And then we went out and found all the samples of fecal samples of all the animals that had been using these lawns over this year period. And we monitored or estimated the nitrogen and phosphorus content in these as well. And because we had the species-specific nutrient content of the fecal material, and we had measured the amount of fecal material that accumulated over this year period, we could estimate how much nitrogen and phosphorus actually went into these ones. Okay, so this is what we found. So the top row of graphs shows the fecal nutrient data. So that's a data point per dung sample. The bottom is lawn specific, so it's a data point per lawn. And if you just look at the top graphs that shows the fecal nutrients, the first panel is for fecal nitrogen, second panel for fecal phosphorus, and the third panel is fecal nitrogen to phosphorus ratios. And it's plotted against the log of herbivore body size. And you can see that the fecal nitrogen didn't actually change as much as we expected. It was, there's of course a slight decrease, but you can see it's not really particularly significant. But the fecal phosphorus had this very strong decrease with an increase in body size. So the larger animals had much lower phosphorus content in the fecal material. And because of this, you have this increase in nitrogen to phosphorus ratios within the species. And if you look at this bottom graph here, what I've done is, so this is on the y-axis, you've got the average defecated body weight on that lawn for that year. And on the x-axis, you've got the average visibility surrounding that lawn. So the average visibility is, of course, the measure of risk. So what this is showing is that where the visibility was high, so in this region here, high visibility, so presumably safe areas, you had generally smaller things eating from there and defecating in that plot. Whereas where the visibility was lower and it was more of a risky environment, you ended up having larger, on average, larger species defecating on those lawns. So just as a slight reminder, if I go back to this little schematic, this is basically what we were seeing here as well, that when you're in the risky side, then the fecal material contribution is predominantly from the larger species. And then what these two last graphs are showing is the nitrogen to phosphorus ratio. The first graph is the nitrogen to phosphorus ratio of the actual fecal input, the total nitrogen to phosphorus ratio input through feces. And the second graph is the nitrogen to phosphorus ratio that we recorded in the grass growing within that, in the leaves of the grass growing within this plot. And it's plotted against the average defecated body weight. So you can see that as the lawn received generally larger or fecal material from generally larger animals, then it was receiving higher nitrogen to phosphorus ratios in the fecal material. And those same lawns also showed higher nitrogen to phosphorus ratios in the grass leaves that were growing in those lawns. Now, of course, this is not the neatest relationship you've ever seen, and we didn't test a cause and consequence. It's just a series of successive correlations. But it does seem to suggest that that the body size of a herbivore could influence the stoichiometric impact of these herbivores. But there's a lot more controlled experimentation necessary before we can really confirm these kinds of relationships. And also because there are at least two other size-related mechanisms that influences stoichiometric impacts of consumers. And the first one being diet. So up to now, we've kind of considered that all the animals are able to find the same diet and they consume kind of the same nitrogen to phosphorus ratios to begin with. And in the system that we were working in Roux and Pelosi Park, this is maybe not such an unreasonable assumption because of these grazing lawns. Because like I was saying, these grazing lawns attract species from, impala side to rhino side. And they really, when the lawns are actively growing, you find these species really actively focusing their foraging effort on these lawns. And they go from lawn to lawn to lawn. And sometimes you might even see these kind of lawn pathways that leads from one lawn to the next. And the lawns, especially if you are in the same area, they often consist of the same species. And they're often dominated by one particular lawn grass species. And for the smaller lawns, it could even be the same individual because it's just growing somniferously, right? So I think in this system, it's not an unreasonable assumption to think that they were getting roughly the same diet, and that's probably why we didn't find such a strong nitrogen decrease in the fecal material. But in another situation, you might expect that the smaller animals are able to select better than the larger animals. So we know that larger animals have lower quality diets that are more diluted by fiber. And so they might be getting less nitrogen to start with, or even less phosphorus in the diet to start with. And this is important because there is a very, very strong dietary signal in fecal nutrients. So if the diet was originally low in nitrogen to phosphorus ratios, then the fecal material is going to be low in nitrogen to phosphorus ratios. And if you look at data from other savanna systems, so this is data from Judith Sitters and Harry Alderventerink, and they've been working in savanna systems in Kenya and Tanzania. And they came with data and said, but we don't find the same patterns in our systems. And this is likely partly because the size selective or size specific selection for diet is operating in these systems. So just as a comparison, this is again the data from Roux and Pelosi, and you see in panel B, the middle panel is the phosphorus decrease that we find in fecal material over body size. And with Harry and Judith's data, the middle panel is again the fecal phosphorus, and you don't see the same pattern. And then, so the second mechanism, size-related mechanism that could influence stoichiometric impacts is metabolism. And of course, we did consider metabolism when we were in our expectation of how nitrogen would change with body size, because we were expecting that nitrogen, the requirement of nitrogen would decrease per unit body mass with body size because of metabolism. But phosphorus, of course, is also a really important component of metabolism. And maybe not unreasonable to assume that these very large animals like impalas to rhinos that have such a huge skeletal structure, perhaps the demand for phosphorus is overwhelmingly for the maintenance and buildup of bone. But once you start including smaller animals, then maybe the phosphorus requirement becomes a more important consideration. And in fact, the original hypotheses from Stirner and Elser was this quadratic relationship. So they were saying that at larger body sizes, you would expect animals would require lots of phosphorus for bone, but at smaller body sizes, they have faster growth rates and faster metabolic rates. So they might require a lot more metabolic phosphorus. And of course, again, if they're eating the same diet, you would expect that the opposite is then true for what remains in the fecal material. So again, a quadratic relationship as depicted in this blue line here, and where the smaller and the larger animals, the fecal material from the smaller and the larger animals have lower phosphorus content and higher nitrogen to phosphorus ratios. And this just again to compare to Judith and Harry's data, So there, if you look at their Tanzanian data set, the fecal phosphorus in the Tanzanian data set, you do see this quadratic response. And just to point out that our data set, we had a lot more of a restriction in body size range than they had. Our smallest animal was an impala. Their smallest animal, I think, was a spring hare. So this also speaks to why we're not finding the same patterns. Perhaps our size range was just too restricted to really see this curvature. But it becomes really tricky when you're analyzing these kinds of things, and there's so many aspects that's influencing fecal stoichiometry. Like I said, diet is an important component. So if your herbivore community consists predominantly of grazers or predominantly of browsers, it's going to make a huge difference to what you end up measuring in the fecal material. And like with, again, comparing these data sets, our data set had consisted basically of grazers and mixed feeders. We had this one lonely giraffe that was the only browser that was included in our data set because our system was a grazing system. So of course we can say nothing about changes in stoichiometric demand along body size in browsers. Judith and Harry's data, especially the Tanzanian data set, again leans a lot more towards browsers. So that's maybe also why we're not finding the same patterns. Ruminants might also differ from non-ruminants because they will also have different diets to begin with, and they'll also have different capacities to extract nutrients from the diet. So basically, we came to the conclusion that our individual data sets are just too small and taxonomically restricted to be able to generalize in the way that we've been generalizing. So even though I've been singing Joshua and Pelosi's praises about it being such a diverse system with so many species, turns out they don't have enough species to be asking these kinds of questions, especially if you're a large mammal ecologist that's too stubborn to start including smaller species in what she's looking at. So Judith and I then decided, actually, well, we know so many people that's collecting fecal material in Africa, and not for these stoichiometric questions, but for all kinds of other questions. So we started sending out emails and asking people if they would be willing to contribute their data to a large shared database on African fecal nutrients that we called the Dung Data Depository. And people were so willing, the reception for this was so great. Everybody was willing to contribute, and they went and dug up old data from musky drawers and whatnot. And so at the moment, we have a database that 37 researchers contributed to this database, 39 if you include Judith and myself. And we have over 10,000 data points. Many of these have multiple nutrients. So it's actually over 10,000 fecal samples with multiple nutrients. It represents 48 herbivore species that range in size from Hewitt's red rock rabbit, that's 2.7 kilograms estimate, to a savanna elephant, that's 4,000 kilograms. It's from 10 different countries, a little bit of a cluster in Southern Africa, which reflects Judith and my network. But it also was collected over a variety of different savanna systems that's different in aridity. So from like a very arid system, the Karoo National Park, to very, very mesic and humid systems like the Roux National Park. I don't know if I butchered that pronunciation. I apologize if I did. And we had this amazing master student, Ridvan Jafar, and he really collated all of this data and did the first mixed effects models on it, and the first relatively broad analysis. And he's just finished his master's, very talented student, and he's looking for a PhD. So I'll just leave that information out there in case there's anybody on this call who'd like to find a talented student. And he extracted some of this data for the talk, and this is just the subset of data that contains both nitrogen and phosphorus for the same fecal sample. And this, so I included those predictions from Stern and also just in the corner here for reference. This is the fecal nitrogen over the log of herbivore body size, fecal phosphorus over body size, and the nitrogen to phosphorus ratio over body size. So you can see that you do have this decrease in nitrogen over body size. I see I forgot to add the actual statistical estimates. I apologize for that, but all of these are significant relationships. So there is this decrease in fecal nitrogen that you would expect because of perhaps the selective feeding of smaller animals. And you do see this curvature in the phosphorus content in the feces. And of course, because of that, this same curvature, opposite for the nitrogen to phosphorus ratio. But this is early days, and this needs a lot more detailed analysis. At this point, it's kind of all data in a pot, analyze. Our next steps will be to try and tease all of this apart and kind of stratify along lines of, splitting ruminants and non-ruminants, splitting grazers from browsers, and maybe also splitting between the different savanna sites, so that we could maybe, stratify along rainfall categories, so we can try and minimize that initial variation from diet. Right. So this is actually my last slide. So I've basically shown you what I came to show, but I guess I'll just conclude by saying that I think there's still a lot of questions, and there's a lot of uncertainty in what impact consumers actually have on nutrient landscapes, and particularly in communities that are highly diverse. And so we know that there's a lot of contextual complexity and a lot of nuance that we still need to describe. And we don't know when different species within a community is working synergistically, or when they are counteracting the effects of the other. So we are very far from describing a net effect of consumers on ecosystems. And also, like, how often is that net effect strong enough to even show above and beyond the abiotic background nutrient drivers, right? But by saying that, I think what I'm trying to say is that I think it is a really exciting time for terrestrial zoogeochemistry, because there's all these questions to ask, and there's so much space, and there's at the moment so much momentum amongst ecologists and zoogeochemists to try and build up this understanding of how species-specific traits interact with ecosystem-specific context to shape these nutrient landscapes. And there's a lot of opportunity for cross-ecosystem comparisons to try and get at what are the changes that we're expecting. So the changes that we expect in terms of biodiversity or trophic complexity of the future, whether this is because of extinctions that we expect, or restoration programs like rewilding. What would these changes mean for the nutrient landscapes as we know it today? And I thank you for listening to me.
Thanks a lot. Now we have a little time for questions. Is there a question from someone in the room? Maybe if you can come forward a little bit so that
Oh, yeah, they can hear you. Well, they could hear my coughing. Apologies for that. So certainly they can hear me. One of the things that makes species different in the system is obviously the life history and the traits, like the size and everything, but also the abundance. And how do you disintegrate for that? Like, can you control for the abundance? For instance, in your results when you're assessing the contribution of large and small species?
Well, I suppose yes, you can control for it in the sense that you can. I mean, you would know which species contributed in which abundances and what abundances are out there in the park. So, for example, like with those graphs that I was showing with the rhino and the zebra and the kudu, I forgot to point out that the kudu, the meso browsers hardly contributed at all to the system. But that's of course because I limited to a grazing system, because we were specifically focused on the grazing system. So in that sense, like that, I basically eliminated them from the whole impact, because there are a lot of meso browsers in that system, but they didn't impact that particular scenario. But yes, so I do agree that abundance is going to factor into it, but there's always ways of getting at that, right? We could also, I think with this DDD, the database that we're setting up, if we can get at what are the traits that influence these the stoichiometry of fecal material, then we can stop with this species nonsense, you know, and we just lump them into traits and say, and then that maybe moves a little bit away from this abundance issue. I mean, you would still have that. Does that answer your question?
Yeah, it's just that I think abundance can vary, like inducing presence between sites, but there's also generally a relationship between, like, for instance, body size and abundance.
Yes.
That would be.
Yes, for sure. Right. I don't know if this was in these graphs, but for example, what we did is, I mean, we were working with the biomass per category, right? So when we were looking at how they differed in terms of the impact that they placed on each of these lawns or on that experimental site, we calculated it in terms of kilogram of animal. And then we still found these differences. But of course, there are, I think that's part of this nuance and this contextual complexity that I'm talking about, that it's going to differ from system to system. And depending on which species you've got and which way they're congregating and if they're moving, and you would have a different impact. And not to say that this is not characterizable, but I think it will vary from system to system, and according to general rules, we would need to be a little bit careful in how we generalize.
Sorry, there's a question online, so we're going to switch. So Marjolaine is asking the question on behalf of someone else. So can you? Yes. No. Now you turned off your microphone. You have to turn it on again.
Ah, Marjolaine, c'est toi. Ah, okay. Oui, bonjour. Hello. Yeah, sorry. We are in CVGP here in Montpellier, and we are using a common computer for the whole lab. So, yeah, my question was about predators. Have you seen these predators in your camera traps, and have you seen a kind of an increased density of these predators in open spaces? So you can see. Really, one thing is the landscape of fear that you imagine with your landscape manipulation. The other is, are these predators more active in these areas or not? So there's a kind of a trophic cascade from predators to grazing, etc.
Right, thanks. Good question, and thanks for that. So our setup was not appropriate to really. That's why I keep kind of every time I talk about risk, I try and correct myself and talk about the perception of risk. Because our setup was not perfect to actually measure what the actual risk differences were amongst these plots. Firstly, because the plots were too small. So yes, we did see predators in these plots, but we're talking about 10 by 10 meters. So occasionally they come past. We didn't have a statistically significant capture rate of predators to be able to say, are there more predators in the risky six plots of 10 by 10 than the non-risky 6 plots of 10 by 10. But we had at the same time collars on lions, which are the main predators in the system, and they were definitely within these areas. And there are a lot of other studies out there that show that there's this visibility impact in how animals are responding, particularly for something like a lion that's an ambush predator. Is a really old study that I thought is quite a nice study, because they showed very clearly, they actually measured what is the success rate of lions in catching different herbivore species depending on how close they manage to get before the animal sees them. So I don't know how these people got so much data on, they must have seen so many kill sites, I mean kills in action, but and they clearly showed that there's a very, very steep drop off. The moment that the ambush predator can't get closer, then they can't manage to catch these herbivores. So the relationship is relatively well established, but in other systems. So in this, our study was not very appropriate for actually checking that particular link. But they were definitely in that environment. And from another study that was also published relatively recently, they did show that these kill sites are more frequently in the denser vegetation. So did that answer the question?
Yeah, thank you very much.
Yes, I have a question related to Simon's question. It's for the, you showed that the meso herbivores seem to spend more time in the less risky area. I was wondering if there could be a difference in group behavior between meso and mega herbivores that could explain even more space in big groups.
Yes. So you mean that this congregating versus not congregating is not a predatory response, just they don't want to be all cooped up in a tiny.
Yes.
Yeah. It could be, definitely, especially in our setup where we had this relatively small gaps that we created. So a 10 by 10 meter gap fits one elephant, you know. So definitely, but I would say that. So our data shows actually, even though it wasn't statistically significant, we found them more often in the non-clear plots, in the 10 by 10 meter little gap, than we did in the big one. So those plots that we created, the inner one was 10 by 10 meter, but the bit that we cleared in the surround was 40 by 40 meters. So in the end, that is quite a big gap within a dense savanna matrix. And we still found the mega herbivores more frequently in the uncleared little 10 by 10. So I don't think that was the case. But I guess it's very for sure.
Okay, so there's an entomologist online wanting to ask a question about insects, I guess. Right, Ricardo?
Yes, Luis. I'm here. Yes, I was just wondering about the dung beetle communities, because since they have a quite remarkable role in the uptake of dung nutrients into the soil, I was just wondering if the different ratios between phosphorus and nitrogen and the differences in the mammal communities could actually have a sort of chain effect on the dung beetle communities. And also another curiosity was, since I know many species of dung beetles are actually species specific or so, if it could be a way to test like extinction chains, like there's a species of mammal that gets extinct, and this as a sort of chain effect on the dung beetle communities. It was just to know if you had some hypothesis or data on that.
Right. Interesting question that I don't think I would be able to answer. I don't have any data on that. But yes, that's an interesting direction to follow. Certain. There was some dung beetle research in the park at the time, but I don't remember now if they were definitely not focused on the stoichiometric question. So I think that that is definitely part of that excitement of exciting times for terrestrial zoogeochemistry, because that question is wide open. And I think that, so you remind me of, I did my honors degree way back when on dung beetles, and on this one particular dung beetle called Terrestrial Pseudochemistry, that is the only, I think, flightless dung beetle. And they occur in these very, very dense thicket-type habitats in the southern Cape, but they used to occur much further across South Africa, Southern Africa. I don't actually know in the rest of Africa how large they're. I think it was a Southern African species. I don't remember now. But they are nowadays limited to these little pockets. And that was because of the disappearance of basically elephant from the landscape. But also, why I mention this now is because they are this relatively large and flightless species. And people were saying that this is all because of this habitat that they used to. They had, complete access or exclusive access to the really dense sites. And they were so dependent on elephant dung, because now one of the problems is that because there's no elephant, they're using buffalo or cattle dung, and it clogs up their mouth parts and they die. But so it's this interaction between the loss of this thick habitat and the loss of elephant, and they had this very neat strategy to be the only dung beetle that manages in this thick stuff, because everybody's seen a dung beetle fly. They're like stupid. They keep flying into everything. And this one dung beetle just walked its way to the dung in this thick habitat. And now the combination of the loss of both elephants. So yeah, you just made me think of that like these extinction cascades, where animals are, and particularly insects, become so specific in their strategies, and then once you lose this little bit of nuance, which cascading extinctions will follow. I don't really know if that fit what you're thinking, but you got me in thinking about my honors degree on Fecal. So.
It's quite interesting what you say. So yes, it was what I meant. Thank you.
Okay, there are quite a few questions online, so if there are not questions in the room, I mean, I would have one actually, since I'm here. I was thinking, something that struck me was that you're saying these herbivores are bringing all these nutrients in these more open areas where there's less vegetation. So how do you think this will impact vegetation in these areas, or there's a flow of nutrients between places where they eat and places where they defecate? You know what I mean? Because to me, these places you show at the beginning, they were open, very little vegetation, and then end up having very rich soil. I'm not very.
So they're definitely lower in vegetation biomass, especially if you're considering also tree biomass and woody biomass, because of course that's part of it, right? But so the pictures that I show was very clearly showing this fecal accumulation, but not all of the areas are, because that was kind of also a biosphere around a water hole. But many of these other open areas still has a completely continuous grass layer.
Okay.
And those grass layers, the grass grows really, really fast. So I think in terms of standing biomass, for sure, there's much lower standing biomass in these, but I don't know about total biomass if you consider how much productivity is happening in these sites and how much removal and how much turnover.
Yeah, I think there is a related question online actually. Albert Vasilis asked this question. Vasilis, can you turn on your microphone?
Yes, hi. Thank you for your presentation. I hope you hear me.
Yeah.
Yes, so yeah, it's a bit related. I was wondering what is the effect actually on the plant community? So like to really close the circle, I understand the nutrient landscape changes, but I was trying to wonder whether would that be important for the plants that grow there, or actually the role of the herbivores is mostly about grazing and trampling, and not so much of how they distribute herbivores. So now I'm talking about the perspective of the plant. Right.
No, that's a good question, and certainly one that I'm very keen on getting into. And I haven't worked on that up to this point, but Judith Sitters, one of the people publishing this other data set, she has worked on it from, and she has this. So she checked she basically fertilized grass and fertilized seedlings, tree seedlings, with small herbivores and large herbivore fecal material that were so different in the nitrogen to phosphorus ratios. And the seedlings were nitrogen fixers, and she could show that the nitrogen fixers were not being let me just get this right. The low nitrogen to phosphorus ratios were benefiting the tree seedlings because they were nitrogen fixers. So they were able to just get their own nitrogen from there, and then they were boosted by this higher phosphorus availability, or at least relatively speaking. And so I think that was maybe the in savannas, at least, the obvious expected shift is that you will benefit if you shift the limitation to be a nitrogen limitation, because you're adding phosphorus to the system, then those nitrogen fixers are going to be happy. And then but of course there's a lot more that could happen. We're also talking about overall productivity rates, and I'm sure if you look into it, there's a lot of predictions that you can have about how a community would shift, and also how a community would shift from trees to grass, like broad functional groups. One of the other things that Judith Sitters works on is that she published this paper where she was showing how because the diet is so determined, it's determining the fecal stoichiometry. She was showing how the grazers are eating diets with much lower nitrogen to phosphorus ratios, whereas the browsers are eating diets with much higher nitrogen to phosphorus ratios. So then the dung is also reflecting those. And because of that, there's this, so the browsers are indirectly benefiting grass communities, and the grazers are indirectly benefiting tree communities. So, and she was kind of speculating about what kind of, I mean, would that have an impact on tree-grass ratios in savannas? Which, of course, perhaps would maybe in a very localized setting that could happen, not on an Africa-wide scale, but still, there's these interesting questions that's ripe for the picking. And with your other part of the question about what is more dominant, the trampling or the actual removal, so I think that very much depends on the animal, on the herd size, on how frequently they get there, because I think that that trampling effect is oftentimes not so detrimental to the plant as you might expect, especially when you have lots of a very grassy system. I mean, then that's not necessarily, unless it's they're compacting the soil, then of course it changes water content in the soils and so on. But so I would speculate. I would think that the nutrient impact should be relatively prominent. But I'm just saying this because I like these topics, and I'm hoping that it would be.
Okay, so we don't have that much time left, but we can take 2 short questions and answers that are online. So let's see first by Christelle Heli Alom.
Hi, Elizabeth. Sorry, hi, Elizabeth. I was wondering, did you have some fire during your experiment? Because you've been working for several years. And if so, as nitrogen and phosphorus are quite volatile, even though it can burn. So do you think that we could measure the depletion and on a long-term perspective as well?
Yes, definitely. Also, very good question. Of course, fire plays an important role in these systems, and it's going to have an immense influence on nutrients, but also on the stoichiometric ratios, because nitrogen is so much more volatile than phosphorus. So, and because of this, for our work with the experiment, we were worried that we're going to get fire impact, and then it's just going to completely ruin all the visibility that we created, and also the nutrients that we're following. So what we did is we burnt all of them from the start. So when we set up these systems, I had a team of about 50 firemen come out and just set fire to the savanna and then stop it again on the other side of my experiment. So in that sense, it did not have an influence on this data, but in practice in savannas, yes, that's part of that intense, like that contextual complexity that we need to come to grips with. And the other thing I wanted to say is that the grazing lawns are too short-statured to burn. So they never burn. So in that part of the study also fire was excluded, but not necessarily from the region. So where it could have an influence is in terms of how the surrounding areas and how it attracted animals and so on. But our nutrient measurements should not have been fire impacted, unless you have deposition of atmospheric deposition from a fire just along the road. But yeah, I hope not, I guess.
Even during a La Nina year, for instance, where you receive a lot of water, rain, maybe those lawns are a little bit more fueled, I would say.
I won't say that it's impossible for them to burn, but from my experience, those lawns, the faster they grow, the faster things eat them. So they're really maintained very, very short and in very green. So when it's not fire season, and these animals are just maintaining them. Then when the fire season comes, or in between the wet season and the fire season, is the season of probably the intense limitation for herbivores. So now they are forced to eat somewhere else, but they keep coming back to the lawns to see, is there a little bit more that I can get here? So they eat those lawns short, long after the lawns stop growing. So I think not. But of course, the lawns are not eternal. There are times when they grow out, and then perhaps, you know, for whichever reason the animals are not coming fast, then of course they can burn. And the other interesting interaction with these lawns and fire is that if you do have a lot of fire and a lot of regrowth after the fire, it pulls the animals up off the lawns, because now that they spread out their grazing, because they're not so dependent on the lawns anymore. And then you could start losing the lawns because you don't have the grazing pressure. So yes, there are situations where they will burn, but not if they are maintained.
Thank you.